Mechanisms of fertilization: sperm capacitation, acrosomal reaction, fusion of sperm and egg membranes. Fertilization is the process in which a sperm cell from a male merges with an egg oocyte from a female to form a zygote. This complex process involves several distinct stages, each essential for successful fertilization. The three critical mechanisms of fertilization are sperm capacitation, the acrosomal reaction, and the fusion of sperm and egg membranes. Let's break down each mechanism. 1. Sperm capacitation. Capacitation refers to the biochemical and physiological changes sperm undergo after ejaculation and during their journey through the female reproductive tract. These changes are necessary for sperm to gain the ability to fertilize an egg. Key aspects of capacitation. Location. Capacitation occurs in the female reproductive tract, specifically in the uterus and fallopian tubes, after the sperm have been deposited in the female genital tract during intercourse. Changes. During capacitation, removal of cholesterol. The sperm's plasma membrane undergoes changes where cholesterol is removed. This makes the membrane more fluid, allowing it to undergo necessary changes later in the fertilization process. Ion changes. Calcium and sodium ions enter the sperm, triggering biochemical pathways that increase sperm motility. Hyperactivation. As a result of capacitation, sperm undergo hyperactivation, a significant increase in motility. This allows the sperm to swim more vigorously and navigate through the female reproductive tract toward the egg. Increased membrane fluidity. The changes to the sperm membrane allow for easier fusion with the egg during fertilization. Functional outcome. After capacitation, sperm acquire the ability to undergo the acrosomal reaction, which is necessary for penetrating the egg's protective layers. 2. The acrosomal reaction. The acrosomal reaction is a crucial step that occurs when sperm come into contact with the outer layer of the egg, specifically the zona pellucida. This reaction allows the sperm to penetrate the egg's protective barriers and initiate fertilization. Key aspects of the acrosomal reaction. Acrosome. The acrosome is a cap-like structure located at the front of the sperm head, containing enzymes that help the sperm penetrate the egg's protective layers. Initiation of the reaction. The acrosomal reaction is triggered when the sperm binds to specific receptors on the egg's zona pellucida, the glycoprotein layer surrounding the egg. Enzyme release. Upon binding to the zona pellucida, the sperm's acrosome undergoes a fusion event with the sperm plasma membrane, releasing enzymes such as acrosin, hyaluronidase, and neuraminidase. These enzymes help digest and break down the zona pellucida, allowing the sperm to penetrate the layer. Penetration. As the sperm moves through the zona pellucida, it reaches the perivitelline space, the area between the zona pellucida and the egg's plasma membrane. At this stage, the sperm head comes into contact with the egg's membrane. 3. Fusion of sperm and egg membranes. The final step in fertilization involves the actual fusion of the sperm and egg membranes. This step allows the sperm's genetic material to enter the egg, resulting in the formation of a zygote. Key aspects of membrane fusion. Sperm-egg. Binding. After the sperm has penetrated the zona pellucida, the sperm head binds to the egg's plasma membrane. This binding is facilitated by specific proteins on both the sperm and egg membranes, including isomo 1 on sperm and Juno on the egg. Membrane fusion. Once the sperm binds to the egg, the membranes of the sperm and egg undergo fusion. This process involves the sperm's plasma membrane fusing with the egg's plasma membrane, allowing the sperm's genetic material DNA to enter the egg. The cortical reaction is triggered at this point, which involves the egg releasing calcium ions. This results in a structural change in the zona pellucida, causing it to harden and prevent additional sperm from binding and penetrating the egg. This is known as the block to polyspermy. Entry of sperm. DNA, the sperm head, now devoid of its tail, enters the egg. The nucleus of the sperm, containing half of the genetic material, or 23 chromosomes, combines with the egg's nucleus, also containing 23 chromosomes, forming the diploid zygote with a full set of chromosomes, 46 chromosomes in total. Summary of the key mechanisms of fertilization. Mechanism. Description. Sperm capacitation. Biochemical changes in sperm within the female tract that enable sperm to become capable of fertilizing the egg. Acrosomal reaction. The release of digestive enzymes from the sperm's acrosome to allow sperm to penetrate the egg's zona pellucida. Fusion of sperm and egg membranes. The sperm membrane fuses with the egg membrane, allowing sperm DNA to enter the egg and form a zygote. Conclusion. Fertilization is a highly orchestrated process involving several steps that are essential for the successful merging of genetic material from the sperm and egg. Sperm capacitation, the acrosomal reaction, and the fusion of sperm and egg membranes are all critical mechanisms that ensure sperm can reach and fertilize the egg. These processes help ensure genetic diversity, successful embryo development, and the initiation of pregnancy. Understanding these mechanisms is crucial for comprehending fertility, reproduction, and assisted reproductive technologies such as in vitro fertilization, IVF.